Hello students, in this lecture we are going to discuss in detail the phase contrast microscopy. This phase contrast microscopy it was discovered by the scientist Fritz Zernike, and for that he got the Nobel Prize for Physics in 1953. In this microscopy we are going to discuss principle, ray diagram, working and applications one by one. Principle of phase contrast microscopy. The light rays move at different speeds through materials having different refractive indices. Some of the light waves are in phase and some are out of phase. When these out of phase rays are brought together, contrast is generated due to interference. सो व्हाट इज़ द प्रिंसिपल ऑफ दिस फेस कॉन्ट्रास्ट माइक्रोस्कोपी कि जब हम फेस कॉन्ट्रास्ट माइक्रोस्कोपी में सैंपल देखते हैं तो वो सैंपल का जो डिफरेंट पोर्शन है, डिफरेंट पार्ट्स है, उनके रिफ्रैक्टिव इंडेक्स कैसे होंगे, डिफरेंट होंगे. तो ये डिफरेंट रिफ्रैक्टिव इंडेक्स होने की वजह से लाइट वेव्स कैसे मूव होते हैं, डिफरेंट स्पीड से मूव होंगे. दैट्स वाई सम ऑफ द लाइट वेव्स आर इन phase and some of the light waves are out of phase, and when the out of phase rays are brought together, contrast is generated in the image due to the phenomena of interference. That is the principle of phase contrast microscopy. Ray diagram of phase contrast microscopy. Here we can see the light source, then condenser lens. Below this condenser lens there is a annular diaphragm which is also called as a condenser annulus. Then here we can see the specimen stage, then objective lens. So this objective lens, it consists of a phase plate which is also called as a diffraction plate, and finally we see the image plane. So here the image is observed through the eyepiece lens. So how this phase contrast microscope works? So light passes from the light source towards the condenser lens. Below the condenser lens we know that here is a annular diaphragm. So this annular diaphragm have a annular ring, and through this ring the light passes through the condenser towards the specimen. So this specimen, it have a different thickness and different physical properties, and different parts of the specimen they have different refractive index, and therefore light moves from this specimen at different velocity and get entered towards the objective lens and from this diffraction plate. So this diffraction plate, it also have the grooves, and the diffracted and undiffracted light get pass from this objective lens towards the eyepiece lens, and finally we see the image of the object. Here remember that the diffracted and undiffracted light, they are brought together to produce the final image of the object which is observed through the eyepiece lens. So this is all about the working and ray diagram of the phase contrast microscopy. Working of the phase contrast microscopy, we already discussed the working of phase contrast microscopy along with ray diagram. Here I have given some points that we are going to discuss. As rays pass from the light source through the specimen, their velocity may be altered by differences in the thickness and physical properties of various portions of the specimens. जब light specimens through pass होती है, at that time the velocity of the light gets changed. Why gets changed? Because this specimen, it have different refractive index, and different parts of the specimen have the different thickness and different physical properties, and therefore their refractive index is also different, and when these light rays pass through the specimen, their velocity gets changed. Light rays passing through the specimen are diffracted differently and travel different pathways to reach the eye of the viewer, and finally we observe the image of the object through the eyepiece of the microscope, and these phase differences are seen through the microscope as different degrees of brightness. ये जो light rays हैं जो differently travel हो रहे हैं यहाँ पे, और they will diffracted differently, they will scatter differently, and here the diffracted and undiffracted rays together form the final image of the object, and therefore phase difference is formed in phase contrast microscopy, and we can observe the bright image against the dark background. A phase contrast microscope uses a special condenser that contains an annular diaphragm. So this annular diaphragm is ring shape, and we know that below condenser there is a ring shaped annular diaphragm. The diaphragm allows a ring of light to pass through the condenser, and that light then focused on the specimen. From specimen it is passed towards the objective lens, and this objective lens it consists of a diffraction plate, which is also called as a phase plate. The diffracted and undiffracted rays are then brought in phase with each other to produce the image that is seen through the eyepiece lens of the microscope. So here is a annular diaphragm of the condenser. So below condenser, this annular diaphragm, it passes the light rays. So light rays get entered into the condenser. From condenser they gets entered towards the specimen. So we know that this specimen have the different refractive indices, and that's why that from different portions of the specimen light travel by different velocity, and then the diffracted and undiffracted light enters towards the phase plate of the objective lens, and therefore the diffracted and undiffracted rays are brought together in phase to produce the final image of the object, which is seen through the eyepiece of the microscope. Now see the images of bacteria which are observed in phase contrast microscopy. So we can observe clearly here the different degrees of brightness and darkness in phase contrast microscope, and also the size of the cell is given here. So due to the discovery of phase contrast microscopy, it has been made possible to accurately measure the size of different types of microorganisms. Thank you. If you like my video, please share and subscribe my channel.